Hello, this is part 2 of chapter 1, corresponding to section 1.3 in the textbook. At the end of this video, you should be able to differentiate between inductive and hypothetico-deductive methods of study, describe how to design an experiment to get objective and reliable results, and to explain what hypotheses, facts, laws, and theories are in science. In the late 1500s and early 1600s, there were two philosophers. Bacon lived in England and Descartes lived in France. And they reframed science as a creative and objective process. Sometimes they worked together, sometimes they worked in competition, but overall they outlined a new procedure for systematically thinking about science, starting with observable facts and then finding similarities, differences, and trends in those observations. And this was the very beginning of what we now call the scientific method. Francis Bacon in particular was a very strong proponent for use of the inductive method. And we find this method to be very, very useful when we study anatomy in particular. So the main idea is the inductive method is based on making numerous observations until the generalizations and trends in those observations are apparent. The inductive method says we cannot prove a claim beyond all possible refutation. But when repeated observations reliably confirm something and no credible observations falsify the claim, we can consider the observations proof beyond a reasonable doubt. However, all truth in science is tentative and subject to reevaluation if new evidence comes to light. On the other hand, we often rely on the hypothetico-deductive method when we study physiology. In this method, we form a hypothesis, and a hypothesis has to be consistent with the known data, has to be testable, and has to be falsifiable. Let's dive a little deeper into this concept of falsifiable. So specifically, when we start an experiment and we are thinking in terms of the hypothetico-deductive method, we want to think about what evidence we would need in order to prove our hypothesis wrong. For instance, if I make a hypothesis and I say that tomatoes will produce more red ripe tomatoes if they're given fertilizer, that is falsifiable. If I give the tomatoes the fertilizer and we actually see a decrease in tomato production, my hypothesis would be falsified. Here's an example of a hypothesis that is not falsifiable. Okay? If I claim that Thor and Zeus are literal corporal entities that are controlling humans' fates and destinies, there's not only no way to show that's true, there's no way to prove that's false. It is not falsifiable. Okay, so very important key here is that falsifiability is a component of the hypothetico-deductive method of thinking about scientific concepts. Now we're going to move into talking about experimental design, how we can plan a good experiment that will allow us to draw meaningful conclusions at the end of our research. The first thing that we need to consider is our sample size, or the number of subjects that we're involving in the research process. So the first reason that we need to consider sample size is to control for the random variation between individuals. We can see just looking around us that individuals have a wide variety of traits, and some of those traits may influence the thing that we're studying. So having a large sample size will minimize the impact that random variation between individuals has. Also, sample size that's appropriate will allow us to be more confident in the conclusions that we draw. When my lab designs experiments that involve bone marrow transplants on mice. The first thing that we do is some statistical analysis that tells us the minimum sample size that we can have that will allow us to draw conclusions. We don't want to waste time and resources on making a study bigger than is necessary, but we also don't want to have a sample size so small that our conclusions are impacted in a negative way. Next, we want to have a control group that is very similar to the treatment group in all the ways that are relevant to the study, except the control group is not going to receive whatever the treatment is. So in the case of the research I do with mice, we try very hard to make the control mice to be littermates to the group that's receiving treatment. And that way the mice are the same age, they have very similar genetics, they grew up in a very similar environment with the same access to food and water. And that allows us to make really meaningful comparisons between the control group and the treatment group and really see the impacts of the treatments that we're investigating. A really fascinating thing that we need to consider is psychosomatic effects. One of the most interesting studies I've ever read about was a study that was done with a group of young adults where they gave all the people participating non-alcoholic beer, but led them to believe that it contained alcohol. And surprisingly, all of the subjects acted as if they were intoxicated and fully believed that they were intoxicated. So really and truly, what we believe, our state of mind, can really impact our physiology. Another really neat example of this is with treating pediatric cancer patients. If a patient is led through a series of visualizations where they imagine the chemotherapy drugs attacking and destroying cancer cells the way that one might see in a video game, the cancer patients have less side effects from the chemotherapy drugs and overall better outcomes. The patient's state of mind has an impact on how well the chemotherapy drugs work. Another element of psychosomatic effects involves the use of a placebo given to the control group. Okay, so a placebo is something that looks like the medicine but has no physiological impact. So if the medicine is in the form of the pill, it would be, sometimes we say a sugar pill or a pill that's just made of nothing. There's no active ingredient in it. If it's an injection, it would be an injection of saline instead of whatever the medicine is that's being investigated. Because people really believe medicine makes you better. And so, if we didn't use a placebo and the control group knew they were the control group, they are likely to be impacted by that due to the psychosomatic effects. Experimenter bias is a little bit different, and if you've ever heard of a double-blind study, that's what this is about. So let's imagine for a second that I'm a researcher and I have developed the next best medication. It's gonna solve everyone's problems. Everyone is gonna be healthy and skinny and happy all at the same time if they take the pink pill that I have developed. And I want to control for psychosomatic effects. So I also am going to give the control group a placebo. It will be a white pill. And I am the researcher. I'm going to meet with each one of the patients in the trial, and I'm going to explain to them what they need to do. They need to take their pill every day, and they're going to record how happy and healthy and skinny they get. And it's very possible that because it's my study and I believe in the power of the medicine that I am researching, that my tone and my words and my body language may give away what the real medicine is and what the placebo is to the patients. Okay, so that would not be a double-blind study. Okay, what we would want to do in a well-designed double-blind study, if I'm the researcher, I would want all the pills that the participants are going to take to look exactly the same. And I would code them with a number, and I would give them to a physician who does not know what my number coding system means. That physician would meet with the patients and explain that they need to take the pills every day. And as they gave the pills to the patient, they would record the numbers in my coding system. And later, when we match up the symptoms of the disease or the progress of the disease with the pills the participants were given, then and only then can we match up what was real and what was the placebo. So in a double-blind study, the patients don't know if they're getting the real treatment or the fake treatment. And also, the person interacting with the patients doesn't know what the real treatment is versus what the placebo is. It's a little bit of a complicated subject there. And the final one is statistics. We want to use appropriate statistical testing to decide if any differences that we saw between the treatment group and the control group were based on actual differences in the treatment, or were they due to random chance. Our statistical testing is going to be the result of the size of the differences we observe between the treatment group and the control group, and our overall sample size. Next, let's talk about the peer review process. Guys, I can tell you the peer review process is a tough process. Other researchers anonymously critique a person's work and give feedback. A lot of research is rejected from ever being published if it's low quality and the peer reviewers don't think it meets the standard of the scientific community. The peer review process is not a one-step process. So in peer review, article is submitted to an academic journal and is given to usually 3 other researchers who give feedback. And a lot of times the original researcher is given the chance to either clarify their work by improving their writing or to do additional experiments that would better support or add support to the conclusions that they're trying to make in their research statements. So this slide here is the most difficult one out of this video. We need to be able to differentiate between facts, laws, and theories as we use these terms in science. And this is not necessarily the way we use these terms in everyday conversation, and that's where the confusion comes in. So first, a fact is something that can be independently verified by somebody who has the proper training. It's just a statement of a simple observation. For instance, a scientific fact could be iron deficiency causes anemia. That's an example of a scientific fact. Okay, so when we think of a law, we usually think of a rule that we shouldn't break or a rule that we can't break. That's how we use the word law in our normal everyday interactions. It's a bit different in science. So a law is a generalization about how matter or energy behaves when we talk about a scientific law. So for instance, DNA base pairing always happens the same way. Adenine and thymine pair together and cytosine and guanine pair together. That is a law of nature. After lots and lots and lots of observations, that is what we have found to be true. Also, if you ever see a mathematical formula is almost always a law of nature. So if you remember from chemistry class, the volume of a gas is inversely proportional to the pressure under given conditions, okay? And we can write that as volume equals 1 over pressure. That is a law. Mathematical formula, think law. All right, now the one that trips everybody up. This one is tricky. Theory. Okay, when we use theory in everyday conversation, we generally mean it like the word hypothesis. I have a theory about why my car won't start. I have a theory about why my sister broke up with her boyfriend. In everyday conversation, theory means educated guess, right? In science, theory has a very, very different meaning. In science, a theory is what we come to when we put together all of the facts, all of the laws, all of the confirmed hypotheses, all of the observations. When we systematically study all those individual pieces of the puzzle and put them together into a unified picture, that is a scientific theory. Okay, so a scientific theory is very different from a hypothesis, very different from how we use the word in everyday conversation. The obvious example of a theory is the theory of evolution. Okay, the theory of evolution comes from thousands of observations, thousands of confirmed hypotheses. We can look at many, many different scientific facts. That's independently verifiable information. Gravity is another example of a theory. We look at all of the observations about how quickly things fall under different conditions and with different angles, and we come up with the theory of gravity. Another example of a theory, the interactions between protons, neutrons, and electrons in the core of an atom. That is a theory. It is the result of our accumulated knowledge of observations and hypotheses and laws and facts. And with that, we come to the conclusion of section 1.3.